Well, it's now 4 o'clock, and I want to be respectful of everybody's time. So let me be the first to welcome you to our series on the microbiome. I'm Francisco Rodriguez with Supplement Innovator. Just to give you a little bit of details on Supplement Innovator, we are a full-service supplement consulting firm. We basically help brands from ideation to formulation develop new products. And we also train raw material sales reps and help raw material businesses overall. We started this series because it fits into a category that I call special projects. And when we see an opportunity to catalyze in the industry new innovations in the world of science or discovery, we'd like to be able to do that. And with the microbiome, I believe it's the kind of undiscovered ocean of opportunity for the supplement industry and the nutrition industry as a whole. And so we started this series last week with our first one. We just kind of gave an overview and introduction, and we were joined by a great presenter, which I'll introduce here in a little bit. And this week we want to talk about the benefits of the microbiome to the nutrition and supplement industries. I think you'll enjoy this presentation. And to reintroduce you to last week's presenter, Second Genome has allowed us to have one of their team members present for us. And Second Genome is a company that brings microbiome science to the discovery and development of therapeutic products. The company has established a pipeline of microbiome modulators that impact infection, immunity, and metabolic disease. Second Genome's development pipeline is fueled by novel technologies for identifying, screening, and scientifically validating product candidates and microbial biomarkers. And Second Genome's technologies have been rigorously validated through partnerships with leading pharmaceutical companies, nutrition companies, as well as academic and government institutions. They have some great news on their website, which you can go to Second Genome, second spelled all the way out, SECONDGENOME.com, and you can look at their news. Some great work that they're doing. They are doing work beyond that in the supplement and nutrition industry, and so we want to make sure that they are well known to the context that Supplement Innovator has. And we have the privilege of having Take Ogawa, who is the Director of Research Services at Second Genome. He spent the last 15 years in the life sciences industry developing and promoting genomics tools and applications. Take was part of the development team that launched the first throughput sequencer at Applied Biosystems, which was responsible for sequencing the first human genome. He also promoted the first next-generation sequencing platform at 454 Roche. According to Take, he's seen the industry shift the research focus from looking at just changes in the human genome to also looking at how we interact with the environment in and around us. So without further ado, I am going to pass this webinar over to Take, and he is going to take you through a fantastic presentation. Take, are you there?
Yes, Francisco. Thank you for the kind introduction.
Absolutely. I'm going to go on mute, and you will be able to share your presentation with us. Thanks.
Great. Well, welcome everybody. I'm excited to talk about the next phase here in our webinar series on the topic of the microbiome. So as I get set up here, just go ahead and do this. Okay. So today's topic, as Francisco described, is we'll be covering the benefits of the microbiome to the nutrition and supplement industry. And in today's discussion, I'd like to share some real use cases, some real recent studies that have come out in the space, in the microbiome research space, where they have found clear correlations, clear associations between our diet and our microbiome. So as a matter of introduction, I'd like to first review what we earlier discussed in the first webinar as to the concept of the microbiome and how we define it. So the microbiome, again, is a term we define as the microbial community that resides in a symbiotic way with a host system. So in our case, that would be the human body, and of course within the human body there are many different ecosystems, from the system within our oral cavity in our mouth to our skin, to our various reproductive systems, as well as our GI system, particularly our intestines and our colon. So as this picture depicts, depending on which ecosystem you look at, you'll find that there's a different commensal microbial community that resides there. And of those, the most complex is in our gut, in our intestines, particularly in the colon. And we have found thus far that there are over 1,000 different species of bacteria that reside in this system in a dynamic way. And so we want to, again, dig deeper into what's happening there and how it relates to our diet and what we in our dietary intake. So just a review in terms of how the microbiome interacts in the context of the human gut is that it plays many roles. And so, of course, as I said, there are thousands of different species of bacteria that reside within our gut, and what you're seeing here is a zoomed-in picture of the various layers of our gut there. And, so if we were to think of this as a layer within the colon, you'll see that there's different cells, our own cells, as well as different bacteria that coexist here. And there's also a very thick mucosal layer that acts as a barrier between the bacteria and our own cells. But in this dynamic symbiotic community here, ecosystem here, again, the bacteria plays a very important role in that it has, of course, protective functions to prevent different pathogens from taking over. It also has a fortifying effect in assuring that our GI system has a structural integrity. But what I want to focus on today in particular is how these bacterial communities, the microbiome, plays a major role in metabolic function. And so here, highlighted in this red box here, are various lists, a list of various functions that the microbial community plays a major role in, specifically around synthesis of vitamins, through the fermentation of dietary components, the production of short-chain fatty acids, which is a major energy source for us. And so these are just, and of course, metabolic pathways. And so these are just some known examples of the microbiome's role in metabolic function as it pertains to the gut. And when we dig a little deeper here and we look for specific examples, of course, our diet plays a role within our gut in several ways. One is more direct in that it has a direct, the diet and the foods that we consume and the various components of the food that we consume has a direct role in how it is that interplay with our gut. But then there's also an indirect role by certain foods and supplements that we ingest actually affects the bacteria that reside within our gut. So it has a direct impact on the bacteria, thus an indirect secondary impact into our gut itself. And so in that, the various symptoms that we might see, the various reactions that we might see are just systemic inflammation in the form of various inflammatory bowel disease, as an example, allergy and autoimmune disorders, and of course increasing susceptibility for things, various infections and even cancer. And so it is a complex system, of course, but no doubt our diet plays a major role, and our gut microbiome plays a major role in our health state, or rather our disease state. So to highlight that case, we look at data that's been collected by the CDC here in obesity trends among U.S. adults. And so we can see that for the last several decades, that the counts of obesity cases have dramatically increased from the 80s and up until where we are today, and then going on beyond. So the number of obesity cases continue to increase, and as a nation, on average, we are at about 20 to 30% obesity rate already. And so why is that? And how is the gut microbiome at play there? So to dig a little deeper there, first we need to build some foundation of where does the gut microbiome come from. So this was a study done by Yasenenko et al. At the Washington University in St. Louis, published in the Journal of Nature in 2012. What they did here, what we are seeing here in the top graph, is three very geographically distinct populations: the Malawians, the Amerindians, and a U.S. population. And they followed a population of infants across time to look at the diversity in their gut microbiome. And so what you're seeing there, again, the top graph here in the y-axis is what's highlighted here as observed OTUs, is basically the count of different species of bacteria that they can find within a sample. So each dot here represents a separate sample, a separate individual. And as we track them over time, we see that with time, the diversity of the gut microbiome does increase with age. And as we zoom in, which is depicted here in the graph here in B, highlighted as B, is when we only look at ages from 0 to 3, that there is significant variability in that diversity, basically an increase in diversity as the infant matures. But by the time they reach the age of 3, it basically stabilizes for the remainder of the adulthood, with increased diversity over a gradual increase in diversity over time. But it's interesting to note here that, of course, across different populations of humans, geographically distinct, ethnically distinct, that there's this general trend that basically our gut microbiome is seeded from the time we're born and continues to increase in diversity to a point where it plateaus around age 3 or so, and then it starts to stabilize for the remainder of our lives. Now, what's also interesting, an interesting finding from this study, as depicted here in this chart in C, is that they found statistically significant differences between the American, the U.S. Gut microbiome diversity with the rest, with the other two geographically distinct populations. And so what's highlighted here with the little stars is basically that there was statistically significant difference in the population as a sum for American U.S.-based people to other parts of the world. So it'll be interesting to see how this trends with, say, other more modern populations, say in Europe or Asia and such. But it is interesting to note that our microbiome here is very distinct. So why is that? So how is it that our microbiome continues to change? So in a separate study here, published in 2011 in the Journal of PNAS, they did a similar study, but now in a single subject, where they tracked that single subject's fecal biome, which is a proxy, of course, of the gut microbiome, over time for several years here, to see how it changed over time and what events in that toddler's life, in that child's life, triggered certain changes in the microbiome. So first, as depicted here as A, what we're seeing here in these little black lines is in the x-axis here are different days within the life of this child where samples were collected, and the different black lines reflect different taxa, that is, different species of organisms that are represented within that sample. So again, as shown in the previous study, is that as we go across time, now this is in a single individual, that again, the diversity and the makeup of those of that gut microbiome changes over time and increases in diversity over time relative to time point 0. As we track the black lines, we see again that the diversity increases, and again the makeup changes over time as well. And to depict that in a little more simplistic fashion, what's highlighted here in the blue bar graph, depicted here as B, is a summary of the diversity there. So each color represents a separate phylum, a separate category of bacteria. And again, as we go across time here, we see that what used to be mainly blue starts to change into different organisms. The makeup of the gut microbiome starts to shift into different organisms, and we're actually seeing what's highlighted here in red. So a different organism is coming to surface as we go across time. And then what's highlighted here in C are different events within that child's life where we see significant shifts, triggers of shifts in the gut microbiome. So as a typical child would, they start on the breast milk liquid diet. Then there are different life events such as a fever, solid foods, antibiotics that trigger certain events. And so if we apply statistics to see what are the general trends in the microbiome, that's highlighted here as step 1, step 2, step 3, step 4. So these different steps are, within a step, are statistically similar to one another in its community, the bacterial community makeup. And as we go across time, we see that there's these different phases, which is, if you just eyeball the data here in the blue, you can see those trends in the microbial community makeup across time, and that certain events seem to trigger or correlate with that. So this is, again, just one individual, but we can expect to see similar trends in all of our gut microbiomes as they mature from the time we're born. Here's an example of a study where we looked at diet effects in the gut microbiome specific to a disease state. So in this case, we were looking at inflammatory bowel disease, which is highlighted here as IBD. So these are diseases like ulcerative colitis, Crohn's disease are categorized in the under the IBD category. And in this study, they took a select number of patients with IBD and put them on very controlled diets. So what's shown here in A are effects of the changes in the microbiome, shifts in the microbiome of individuals. These are patients that were put on LRD. These are low residual diet, a diet which are basically low in fiber, which have been shown to be effective in patients of IBD. And then in category B here is SCD, which are specific carbohydrate diets, so another type of diet that's been shown to be effective in IBD patients. And in the same patients across a trial with a washout period, they tried these two different diets in the same patient. So you can see patient IBD1, IBD2, IBD4 in category on diet B here is the same patients, IBD1, IBD4, pre and post. So to see how the diet impacted these patients pre and post based on this diet, and you can see, oops, I'm sorry about that. Let me go back. You can see here, and this is a heat map that you're seeing here, so the different vertical lines, the different colors reflect changes in the microbiome that we're seeing. And summarized there, we see that based on the low residual diet, that 11 species were found to be increased based on this diet across different patients, and certain species, 13 species, were shown to be decreased based on this diet across patients. And then when we look at this other diet, the SCD diet, we see that it had even more of a dramatic effect, where 134 species were shown to be increased, 6 species were shown to be decreased. So no doubt, in a very controlled experiment, a very controlled trial, we're seeing that there are meaningful changes to our gut microbiome based on these controlled diets, even in the context of a disease, a patient. Here's yet another control study where now it's a little more, the hypothesis was a little more focused on healthy individuals, but on a modern diet phenomena, which in this case they were testing artificial sweeteners that are found in, of course, many of our packaged products, such as beverages. And so when we looked at the effect of the artificial sweetener on the gut microbiome, what you're seeing here is what's called a principal coordinate analysis chart. So each of these dots represents a separate individual's microbial signature, and the distance between these dots represents how different they are. So the further away they are, the more different they are. And so we can see that based on a glucose intolerance response, and we color that as responders versus non-responders, the red dots, who were the responders, versus the black dots, who were the non-responders, depicted here in this principal coordinate analysis chart, just based on 4 days of being on this diet of artificial sweeteners, that there is a statistically meaningful difference between subjects, healthy subjects, that had a glucose intolerance response versus individuals that had no response, just based on this analysis of their overall microbial, their gut microbiome signature. So then when we go look at, well, what bacteria made up these gut microbiomes, we do see a significant difference between the non-responders and the responders in terms of the types of organisms that reside from pre-treatment to post-treatment, but also when we see the individuals that had a response, that their gut microbiome actually changed based on this new diet, this new controlled diet of incorporating artificial sweeteners, versus the non-responders, where basically, if you were to just look at this, you would see that the makeup of their gut microbiome did not change, while with the responders it did change. So this is an interesting association that we're seeing between a clinical response, if you will, versus what's happening in the gut. So there's an association there where we need to dig a little deeper to understand the mechanism of that change based on the gut microbiome. Lastly, there is an interesting study here that correlates the role of exercise and diet to the microbiome. So in this particular study that was published last year in the Journal of Gut, they took a team of professional rugby players in Ireland, the All Blacks, and looked at their gut microbiome and how it compared to age-matched male subjects that were considered to be relatively healthy. And so what you're seeing here in the chart in the left here, depicted in black, are the the individual athletes on this professional rugby team compared to the healthy controls, which were divided amongst individuals of relatively high BMI and low BMI. So low BMI is depicted in red, high BMI is depicted as in green there. So what we're showing there is the creatine kinase levels, which is a metabolic marker within the blood, and compared amongst these different individuals, and we do see a noticeable difference that, of course, as suspected, athletes have a much higher metabolism. And what's not shown here is that they also had lower inflammation markers, which correlated again to their dietary lifestyle and their exercise regime, compared to, again, a controlled representation of a normal population. Then when we go and look at the gut microbiome, which is depicted here in the principal coordinate analysis chart here, we see that, of course, the black dots, which depict the gut microbiome signature of the rugby players, the professional athletes, versus a normal signature, which is depicted in the green and red, that they do cluster together in their own distinct way, in their own cluster, if you will, relative to the other populations. So again, being a professional athlete, having a high exercise regime, and the types of diet that they have, which is high, of course, high caloric, but also higher in protein in terms of percentage, tends to shift the gut microbiome in terms of its overall signature and the community that makes that up. So then this, again, shows an association between diet and exercise to the gut microbiome. I'll wrap up there and send it back to Chico, but just to highlight where we are today, we've covered two topics, and as we dive deeper and deeper into the microbiome and its association to diet and supplements, we want to start to build specific stories around the probiotics in the microbiome, other supplements beyond probiotics and its effects on the microbiome, and then finally go into ways in terms of how we can bring microbiome into research that's taking place in this industry. So with that, I'll send it back to Francisco.
Taka, that was fantastic. Thank you for that. I hope everybody enjoyed that. I have a few questions that I posed over in the comments section. So Taka, I thought I'd bring it over to you on some of these questions to see how you might respond. Mm-hmm. So the first question I have is you show it looked like a significant decrease in American gut diversity versus the other groups that were tested. Right. Number one, what are some of the things that would concern you about that? Number two, what would you like to see done to change that? And then what roles could nutrition and supplement companies play in possibly changing that paradigm of decreasing diversity? Right.
All great questions. And so no doubt it is, to answer your first question, it is concerning to me and many others, I'm sure, to find that at least on average in the controlled experiment that we saw, at least in the context of that study, that Americans typically tend to have lower diversity than other population. And that is concerning in that in other studies we have found that lower diversity tends to correlate with various disease states such as obesity, diabetes, other metabolic disorders, inflammatory disease in the gut such as IBD. So again, there has been association of lower diversity meaning a less than ideal health state. So that is concerning to some degree, but of course these are relative, so we have to take that into context. So it does speak to, of course, dietary trends, I would suspect, in the US versus other parts of the world, but also we have to keep in mind that there are other factors that contribute to the diversity of the gut microbiome, such as our genetics. So of course, a lot of our microbiome is seeded by our parents and our familial members, and so there's that aspect of where does our microbiome come from. But even our own genetics, they have shown, so that is our own DNA can affect the types of microbes that cohabitat within our gut. And then the other piece, of course, is medication and drugs. And so we know that in modern countries that we prescribe antibiotics that have a direct impact on the gut microbiome versus other parts of the world that might not have access to the same sort of health care. So we might be seeing some of that impact directly or indirectly in the diversity within our children. So that is something to keep in mind as well.
It's fantastic. Well, I think that this presentation really communicates effectively, hopefully for all of you that were able to listen, the reality that we play a role in the supplement industry and the nutrition industry on gut health and what that means for other health outcomes. So I hope that you'll send this video on, have others join us in future webinars on the microbiome, and if you weren't able to ask a question today, please feel free to shoot a question over to me at francisco@supplementinnovator.com, and we'll be sure to update you as soon as this video is ready so that you can invite others to watch it. And thank you again for joining, and we look forward to the next time on the next webinar. Thanks a lot.